Influenza or the flu is a contagious viral infection that attacks the nose, throat, and lungs. It is characterized by fever, chills, runny nose, sore throat, cough, muscle aches, and fatigue. The flu virus is extremely small and is only visible through electron microscopes. Inside the virus, genetic material contains the information to make more copies of the same virus. A protein shell provides a hard, protective enclosure for the genetic material as the virus travels between people or animals it infects. An outer envelope allows the virus to infect cells by merging with cell outer membranes. Projecting from the envelope are spikes of protein molecules. The flu virus uses its H spikes like a key to get inside the cells. N spikes allow copies of the virus to break away from the infected cells to infect more cells. There are 17 known types of H spikes and 9 types of N spikes that scientists use to name different flu viruses, such as the virus H5N1. Influenza is transmitted by touching an object that has flu virus on it or through exposure to body fluids from people or animals infected with the virus. When an infected person sneezes, droplets carrying the influenza virus may land in one's individual mouth or nose and then move into the lungs. Once inside the body, the influenza virus comes into contact with cells in the nose, throat, or lungs. The H spike on the virus inserts into a receptor molecule on the healthy cell membrane like a key in a lock. This action allows the virus to get inside the cell. Next, the virus travels inside a sac made from the cell membrane to the cell's nucleus. Then, the viral envelope and cell membrane sac combine, allowing the viral genetic material to leave the sac and enter the nucleus. The viral genetic material hijacks the energy and materials in the cell's nucleus to make thousands of copies of itself. Some of the genetic material moves out of the nucleus, then attaches to ribosomes, which are the protein building parts of the cell. Ribosomes use information from the genetic material to make other viral proteins such as the H and N spikes. A packaging structure in the cell called the Golgi apparatus carries the H and N spikes in vesicles which merge with the cell's membrane. All the parts needed to create a new virus gather just beneath the cell's membrane. Then, a new virus begins to burst off from the cell's membrane. During this process, the newly created virus gets stuck on the cell's membrane when the viral H spikes locks onto membrane receptors. However, the virus has a way to get around this problem. The viral N spikes freeze the virus by cutting it away from the receptors. New influenza viruses are now free to infect more of the cells and cause the victim to develop the flu. The best way to protect yourself from the flu is to get the flu vaccine every year. You may receive the vaccine as a shot which contains dead versions of several types of viruses. The vaccine exposes your body to several types of the influenza virus that are too weak to cause infection but just strong enough to stimulate an immune response. Within 2 weeks, cells in your immune system make markers called antibodies which are specific for only types of the flu virus you were exposed to. The antibodies attach to each flu virus and prevent it from attaching to your cells. Antibodies are also able to attach to more than one flu virus, which causes the virus to clump together. Your immune system responds to signals from the antibodies by engulfing and destroying the clumps of viruses. Later, if you are exposed to these same types of flu again, your body recognizes and destroys them so you will not develop the flu from these same viruses. For continued protection against new viruses, you need to get a flu vaccine every year.